Hello everyone, welcome to Pharma class. मैं हूं विशाल and today we are going to learn about PCR, that is polymerase chain reactions. What is this process for? This is for the DNA amplification or amplification technique. What does it mean by DNA amplification? By using a single sequence of DNA or a single DNA, we can make the multiple copies of DNA. This is the process for that we utilized it. So this process is developed by or invented by the Carey Mullis in 1983, and this is the amplification technique in which by using the target DNA or single DNA we can make the multiple copies of DNA. This is short term in vitro process. What does it mean by in vitro process? It is carried out outside the cell or outside the body. So that is why we call it in vitro technique. Now we will talk about the components. What are the components that we utilize for this process? So there are about 6 components. First is DNA polymerase, DNA template. What is DNA template? This is the target DNA or sample DNA that is the means important, and for that we have to make the multiple copies that we call the DNA template. And second one is DNA polymerase enzyme. So there are a number of DNA polymerase enzyme, but here we utilize tag polymerase. What does it mean by tag polymerase? That is Thermus aquaticus. It is isolated from the bacteria and this is thermostable enzyme. So that is why we utilize this tag polymerase, because in this process we increase the temperature for means it is about 95 degrees Celsius. So for stability point of view we utilize this polymerase enzyme. There is another polymerase, that is PFU polymerase, that is Pyrococcus furiosus. This means there are many, but these 2-3 polymerase enzymes we can use for this process. Magnesium 2+ ion as a cofactor. For increasing the activity of DNA polymerase we can add the magnesium ion as a cofactor. Now we'll talk about the primer. Primers are what? These are the short sequence of nucleotides. They have the known sequence that attach to the template, that can attach to the DNA template, provide the starting point for the synthesis of DNA and help to choose the exact portion for amplifications. Means for amplification they need to choose a specific position or specific portions. So primers may be forward primer and reverse primer. So 2 primers, oligopeptide here we utilized. Now we'll talk about the nucleotides. What is nucleotide? These are the building blocks for the DNA synthesis, like adenine, guanine, cytosine, thymine, uracil. Now the next is reaction buffer. For overall reaction there is a requirement of medium, so we utilize buffer, we call it reaction buffer. So if we'll talk about procedure or mechanism, so the polymerase chain reaction has the 3 phase or 3 process. First one is denaturation, second is annealing, and third one is extension. So we will understand what are the difference between these 3 procedures. First we'll talk about denaturation. Denaturation is carried out if we will see in a graph, there is a temperature and time. So it is about, if we'll talk about the temperature, it is about 95 degrees Celsius. So first what we do, we take all the reactants or components to the tube. There is a small tubes taken to the tubes and fit to the apparatus or machine that we call the thermocycler, and we increase the temperature for about 95 degrees Celsius. So this process called increasing temperature for 1 minute for 94 or 95 degrees Celsius, this process is called denaturation. What happened in the denaturation? The double-stranded DNA gets separated into the single-stranded DNA. This is the process what we call denaturation. And then after the separation of DNA, there is another process that we call annealing. Annealing, what happens? We just down the temperature for about 54 degrees Celsius for 45 seconds. So the 2 primers get attached to the DNA template. So what we call the primer hybridization also we call it. They attach to the 3OH end of the DNA template. Now the third process extension started when for the 2 minutes we just make the temperature 72 degrees Celsius. So what happened in extension? The DNA polymerase, after joining to the primer, they attach the different nucleotides or building blocks to the DNA and they just make the copy of DNA. So here we get the 2 copy of DNA in a first cycle. So this process is repeated again and again, we get the multiple copies of DNA via simple in a simple PCR reaction. So that is why we call it chain reaction. Here we utilize polymerase enzyme. That is why we call it the polymerase chain reaction. Now we'll talk about the advantage. So this is rapid, and rapid means it is very fast. Sensitive process means in a very small amount of sample we can get the multiple copies of DNA. Simple, very simple process. Disadvantage, what are the disadvantage? Technical skill means there is a requirement of technical skilled person to running this machine. High equipment cost, high test cost. Now we'll talk about the application of this process used as a diagnostic tool, and genetic and infectious disease identification. Means there are broad utilization of this polymerase chain reaction. We can utilize it into the research field also, in the drug discovery also. So this was all about the PCR, polymerase chain reaction. So thank you and all the best.